All right, our next speaker is Igor Grigoriev from the Joint Genome Institute, and his talk is entitled 1000 Fungal Genomes Project.
Good morning. Thank you for invitation to this workshop. For introduction, I lead Fungal Genomics Program at Joint Genome Institute and coordinate microbial eukaryotic programs. Today I'll talk about 1000 Fungal Genome, one of projects run by Joint Genome Institute. Before I do this, couple words on JGI. So we're funded by Department of Energy. We serve as a genomic user facility. Being located in California near San Francisco, we support a large community of researchers around the world. And we support various projects in areas relevant to DOE mission and bioenergy, carbon cycling, and biogeochemistry. Majority of our projects comes from calls from proposals. One of the largest one is community science program, which is sequencing of different flavors of genomes, transcriptomes, metagenomes, as well as sequencing and metabolomics, up to 10 terabases per project. The mid-range proposals are actually a collaboration between multiple user facility. One of the most known is JGI AMSL, Environmental Molecular Science Lab in Richmond, Washington, where we combined sequencing at JGI with proteomics and microscopy at AMSL. And this year we started a new investigator semi-annual call, one of the most popular for those people who just about to embrace the area of genomics, to start with a small-scale sequencing to produce primary data for larger-scale projects. We focus our efforts on plants, fungi, microbes, which cover bacteria and archaea, and metagenomes. The fungal genomics started at JGI back in 2004 with the first sequenced basidiomycetes, whitewood fungus, Phanerochaetic chrysosporum, very important for Department of Energy because it's capable of completely decomposing a plant biomass. So this year we celebrated the first 10,000 genomes, fungal genomes, in our fungal portal microcosm, which includes genomic data integrated with all types of multi-omics data sets and interactive tool for analysis, functional and comparative genomics. So I assume that not everybody in this room is playing with fungal phylogeny on daily basis, so I will navigate through this tree a little bit. So the group of Agaricomycota include most of the mushrooms. That's most known fungi. Then Uroshiomycetes includes a lot of antibiotics, and the first one, penicillin, was discovered by Alexander Fleming here in London many years ago. And then another important aspect, touristy aspect of London is pubs, of course, and they're connected to Saccharomycotina, which are important for producing bread and all kinds of valuable drinks. And then we enter in the area of early divergent fungi, which among others include the group of ruminant fungi and anaerobic fungi that, as was discovered recently, includes the largest catalog of lignosulfitic enzymes, those that are very important for processing plant biomass. So the 1000 Fungal Genome Project is one of those projects that come to JGI via community science program. The goal, it started back in 2012, led by Joyce Patafora from Oregon State, with the idea to produce a reference genome for every family of fungi. And there is about 500 to 700 families known so far. And here you see the tree with leaves corresponding to classes. And every bar represented diversity within a class on the level of families. You see the most diverse ones, such as Agaricomycetes, which includes mushrooms, as well as the Basidiomycetes, which is the largest group of known plant pathogens. And the color of the portion of the bar correspond the level of genomic information we have for each of these families. So red are those families that have 2 or more reference genomes, for which we assume that the goal of the project is achieved. Yellow has 1 reference, and we're aiming at producing at least 1 more reference, and green corresponds to families that have no sequence genome so far. So total about 700 families. You see that's about 1/3 of them are families for which we don't have any reference genomes. So this is a snapshot of the data that we have today. A few years ago when we just started, we had slightly different balance of the colors. And over these few years, over 200 families evolved from being green to yellow to red. So we launched the second and third phases of the project, realizing that not all the fungi are equal. So the second phase is focused on early divergent fungi, where we developed special methods such as single-cell genomics. And Dicaria also required closer attention because we realized quickly enough that a single reference genome doesn't serve the purpose of exploring diversity, ecological context, and other questions like that. So the 3 primary goals of the project is to produce a reference data set to explore fungal evolution, to build catalog of enzymes, genes, and organisms that can serve biotechnology, and also produce a reference data set to start embracing the challenges of eukaryotic metagenomics. So it started as a project with 12 PIs, and at this point it's evolved to a really large-scale community effort with over 90 labs participating in this project. We developed a set of tools that allows any researcher around the world to nominate species for sequencing. When you click on this fungal tree of life, it has a menu. In addition to a list of sequence genomes, it has a nominate button, which is connected to this list of 700 families that are color-coded. Again, red are 2 or more genomes. Yellow, we have single genome and still accept nominations. And green are the families where there is no genome sequence so far. So at this point, over 90 participants, 800 nominations, 450 genomes sequenced, and 48 of them are published. We're in the progress of analysis of the next couple hundred of sequenced genomes. And the first snapshot of the data is basically a look at evolutionary history of fungi. So when we cover this fungal tree by important periods in the history of Earth and important milestones in biological history, we can see that the origin of fungi about 1 billion years ago, they emerged before the origin of streptophytes. Then all the major phylas evolved prior to Cambrian explosion, and then diversified significantly after the origin of land plants and flowering plants in particular. So, as I said, this project started by Joe Spatafora, and Jason Steitch is focusing on this initial comparative analysis. These days, the first 1,000 genome were sequenced with diversity of sequencing platform, starting from Sanger, going through 454, Illumina, and these days we sequence most of the genomes with PacBio. As you probably know, PacBio gives us benefits, not only decoding the 4 nucleotides, but their modification. One of them is 6-MA methylated nucleotide. And we know that 6-MA is important in restriction modification systems in bacteria. And a couple of years ago, it was rediscovered for eukaryotes on a couple of model organisms. And now that we have sequenced fungal genomes across a tree of life, we can look at 6-MA levels across and notice that the level of 6-MA in fungi, especially in early divergent fungi, much higher than what was known so far at 3% versus 0.5% that was reported before. So they focused in 80 regions. They're symmetric, and they form islands in connection to promoters of actively expressed genes that play an important role in gene regulation. So the next kind of challenge or technical sort of solution that we developed in the course of this project is to address the disbalance between early divergent fungi that includes over half of different known phyla and Dicaria, which concentrate about 80% of sequenced genomes. So for these purposes, we developed single-cell genomics, and we're able to have a first look at this fungal dark matter, early divergent fungi. This is the 7 representative genomes sequenced with this technology. 3 of them represented the first Zergomycotina genome sequence so far. And this is a collaboration with Tim James and Tom Richards. And this is a starting point for us to embrace a broader unculturable diversity of early divergent fungi. To summarize, I hope I'm still within my 12 minutes. So we developed various resources to enable community to run their projects with genomics tools at JGI. Community science program is one of the channels to get into doors of JGI. We have over 1,000 fungal genomes in microcosm right now, and the 1,000 fungal genome project is halfway through. PacBio sequencing is our standard platform for sequencing and enable us to discover 6-MA methylation in fungi. And we also start delivering the first single-cell fungal genomes. To conclude my presentation, we would like to follow to use the existing infrastructure and follow the development that we achieved in the Fungal program to get to the similar scale of sequencing in algae and hopefully in protists in collaboration and close coordination with other groups of JGI. We started both fungal and algal projects back in 2004. When we started official fungal program, we had about 50 sequenced genomes. That's where we are with algal genomics. And algal genomics is a part of our strategic plan. And you cannot look at photosynthetic protists without looking at those that don't have chloroplasts. So to conclude, to explore this diversity of eukaryotic genomics, I also would like to mention the genomics meeting, our JGI user meeting that happens every year, next year happening in San Francisco in April. Thank you for your time, attention, and happy to answer questions.
We have time for a question.
Is this,
Well, let's have Kreith. You see it, just to spread it around.
I was curious about your order of magnitude difference between the genomes that you have sequenced versus those that you actually have published. You have like 450 sequenced, 48 published. Do you have a sort of target timeline from sequence to publish, or what's impacting that difference?
Right. So the publications that I mentioned are only in the context of 1,000 genome project. And it takes on average 2 to 3 years to convert data into attractive story. So right now we're processing the next largest data set of about 300 genomes coming from that project, aiming to have a solid draft early next year. So that will cover a majority of those 450 that we
I
released.
think that's something that we're going to have to talk about. If this EVP is about putting data out for people to use, then we need to worry less about 3 years of gathering a story before we put things out, and more about getting data out.
To clarify, the data becomes publicly available as soon as they're sequenced. So that's DOE policy. It takes us time to convert it into a story, but it's available for everybody else.
Thank you very much.